Good morning, class. Our today's topic is sexual reproduction in flowering plants. Children, we know that reproduction is a vital process without which species cannot survive for a long. And if we are going to look at the diversity of the structure of the inflorescence, flower and floral parts, shows an amazing range of the adaptation to ensure the formation of the end product of the sexual reproduction, that is the fruit and seeds. So in this chapter we are going to deal the morphology and structure and the process of the sexual mode of reproduction that is found in the flowering plants or in the angiosperms. We know that a flower is a very fascinating organ of angiosperm microbe, and it is known as a modified shoot. So we can say the flower is the reproductive structure of the angiosperms. Here is a diagram that shows the longitudinal section of the flower, in which you can see the petals, sepals, and along with the androecium and gynoecium also. The branch of this botany is known as a horticulture, which deals the cultivation of the flowering plants, and the several hormonal and structural changes are initiated which lead to the differentiation and development of the flower primordium. Primordium is actually an organ in its earliest stage of the development. So this is the earliest stage of the development. The foundation for the subsequent development is known as the primordium. Primordium, leaf primordium, flower primordiums, anything the primordium, an organ that it's a earlier stage of the development. Inflorescences are formed with the beard, the floral buds, and then the flower. Means inflorescences are coming first, and after that floral buds are there, and then flowers are there. In the flower, the male and female reproductive structures that we called it androecium in the male and gynoecium in the female, it is differentiated and developed at the reproductive phase of the life of any plant. So there is the pre-fertilization event. In the pre-fertilization events, we have to study about a typical stamen. Stamen is the male reproductive part, the long and the cylindrical stalk which is found inside the flower, and the part of this stamen is known as the filament, the terminal bilobed structure which is called the anther, and it is of two types, monoecious and dioecious. For example, in monoecious, like in hibiscus or China rose, and in the dithecus, it is found in the brinjal. The proximal end of the filament, means the end that is attached to the thalamus or the petals of the flower, it is called the check terminology. Means the number and length of the stamens vary. What is the length and what is the number of the stamens, it is vary in the each and every flower. So here is the picture, that is the anthers and the filament. It is known as a typical stamen in the flowering plant. Angiosperms, anthers, if we are going to see the how many lobes are there, how many bundles are there, how many theca are the lobes, it is known as the theca also. So it is one of them is known as the dithecus. Means anther is bilobed, two lobes is there, with each lobes having the two theca. Means there is the four theca is found. So a longitudinal groups that run lengthwise separating the theca. So theca is going to be separated. Here is the transverse section of the androeciums or the stamen bundles. The anther, microsporangium is there, and you can easily find out the four theca is there, two in each lobe. It is also known as the pollen sac because pollen grains are going to be developed here in this sac. This is the structure section cutting of an structure of an anthers. If we are cutting down it, the centering, you can see it containing vascular bundle, that is the filaments for the conducting tissues responsible for the conduction of the nutrients, and then there is a tetrad structure. If we are going to study this tetrad structures, the outer layer is known as the epidermis, then the fibrous layer is there. In the center it is the tapetum, and in the center you can find the pollen grains, or you can see the pollen mother cell which is diploid in the number. And after this pollen, by the process of meiosis, the tetrads pollen separated and pollen grains are developed. Means from one single pollen mother cells, 4 pollen grains are developed by the process of meiosis cell division. This is the TS of anther. Picture is taken from your book. The very vast structure. Outer layer is the single layer, that is the epidermis, and then endothecum. Endothecum means thecus inside layer. Then the 2 or 3 middle layers is there. Microspore mother cells having the microspores there. The inner layer is known as the tapetum. So it is known as the sporogenous tissues. Sporogenous because it is going to be develop the spores, male spores or anthers, pollen grains. In the dithecous conditions, we know that it is a bilobe nature. Two bundles are there. Two lobes are joined together. The anther is of four-sided, means the tetragonal structure. 4 microsporangia that located at the corners, 2 in each lobe. The microsporangia develop into the pollen sac. What is this pollen sac? They extend the longitudinal and packed with the pollen grains. All the pollen grains are found in the pollen sacs. Structure of a microsporangium, if we are going to study in the transfer section, a typical microsporangia appears circular in the outline. Means it is circular. It is generally surrounded by the 4 wall layer. Now, outermost wall is known as the epidermis, then endothecum, after that middle layers means 2 to 3 layers, and the inner layer that is known as the tapetum. The outer 3 wall layers perform the function of protection. It is only meant for the protection and help in the dehiscence of the anther to release the pollen after the maturity. But the innermost wall layer, which is known as the tapetum, what is this role of this tapetum? It nourishes the developing pollen grain. When the pollen grains are developed inside the developing stage, inside the lobe, the tapetum is responsible to nourish it. And the cells of the, because the cells of the tapetum possess the dense cytoplasm and generally have more than one nucleus. This nucleus divide without the cytoplasmic division because it is known as the polyploidy. Polyploidy means when in a cell there is so many nucleus, more than one nucleus is present in the cytoplasm. Without the cell division, it is known as the polyploidy. So polyploidy is a very common characteristics of the angiospermic plants. In the TS of anther lobe, you can see here after the maturity, all the 3 layers is going to be dehiscence. When the young anther in a group of the compactly arranged homogeneous cells, means similar cells, it is also known as sporogenous cells because spores are going to be developed here. It occupies the center of the each microsporangium. And the cells of the sporogenous tissues, it undergoes the meiotic cell division to form the microspore tetrads. Tetrads means 4 microscope is going to form, or all are the haploidy, means all are the haploid in number. What is the process of this development? This development process is known as the microsporogenesis. Means microsporogenesis is the process in which the cells of the sporogenous tissues going to meiosis cell division. The process is known as the microsporogenesis, and it is responsible to form the 4 cell haploid. They stay together till the anther mature, and after dehydration or dissociation, all the tetrad cells are to be separate. So cells of the sporogenous tissues are known as the potential pollen or the potential microscope mother cell, having the potentiality to generate the pollen grains. And the microspore tetrads is going to develop into the pollen grain. So there are the tetrad cells separates to form the pollen grain. Now, what is the structure of the pollen grains? You can see in the picture, so many structures is there. Many structures like sound, round, oval, and it represent we know that it is represent the male gametophytes. So the shape, size, and colors and designs are found different in the different species of the plant kingdom. The fossils, because it is known as the fossils also. Why it is known as the fossils? Because the presence of the sporopollenin. Sporopollenin is found there, so that's why these pollen grains are also help in the fossilization. And because this presence of this sporopollenin, and sporopollenin, one of the most resistant organic material is known, and the it is found in the outer covering. Actually, the outer covering is known as the exine. The exine is a very hard, and it is made up of the sporopollenin. And what is this responsibility of this material, this resistant, most resistant organic material? And then it can understand the high temperature, strong acid and alkali also. So that's why no enzymes that degrades the sporopollenin is so far known. So it is also help in the fossilization also. And it is a very hard that the pollen grains are well protected from the hazardous environment when they are pollinated by the biotic or abiotic agents. So it is a taxonomical significance or the pattern and design of the pollen grain. So the in the cryo preservation process, which is in the process is minus 196 degree centigrade, it is used in the crop breeding also. So we can put it under the cryopreservation also. The pollen food nutrition values are very important, that it is used in the increase or enhance the performance of athletes and race horses also. Sometimes this pollen grain cause the allergy also in case of the human being also. So, like bronchial infections, sometimes asthma is also there, bronchitis is also there, like through the pollen grain of the parthenium grasses or the carrot grass. A very severe allergy sometimes developed in case in the human being. And the we are going to see the viability of the pollen grains is depend on the temperature and humidity also. Means few pollen grains is developed within a few minutes, and few takes times for several months for the development. In few minutes, the like rice, to few month, it is just like in family Solanaceae, Rosaceae family, Leguminosae family. So it will take a time for the special development. In the inner layer, which is known as the intine wall, it is a thin and continuous and made up of the cellulose and pectin. The cytoplasm is also present in the center, and it is surrounded by the plasma membrane. That pollen grain wall thick wall, exine and intine. Exine thick, hard, very ornamental also, very beautiful also, and it is made up of the sporopollenin, we know that. And then the germ pore is there present in the area where the no sporopollen is present over here. Means circular aperture, it is found in the exine portions, sporopollenin is absent. And what is the responsibility, what is the function of this germ pore? That pollen tube emerges from the germ pores, it is coming out from the germ pore. The inner wall of the pollen grain, that is intine, we know that it is a continuous layer in the made up of the cellulose and pectin. So this is the diagrammatic presentation, means what the substances is there. It is known as the pollen kit also. And in the A type there is a pollen kit, means the fatty acids are deposit in the cavity. We can see it is a sporopollenin, and in the intine cavity, that cellulose, hemicellulose, most of them are very nutritious in the deposition. The if we are going to children, we know that sometimes the storage of this pollen grain for years in that liquid nitrogen or in the minus 196 degree centigrade for the later use actually in the plants breeders. Sometimes it is programs, that is programs developed by the farmers and by the researchers also. These centers for the storage of this pollen pollens are called the pollen bank actually. And if we are going to study the structure and development of this male gametophytes or the pollen grain, you can see here in the picture a germ pore is there, exine and entine is there, very common pollen grain is there that is surrounded by the plasma membrane. And during the development from the microspore mitosis, two cells is there. One is the vegetative cell, that is tube cell also, and another is the generative cell. What is the role of this vegetative cell? That it is a vigor, has abundant and responsible for the food reservoir, and a large irregular shaped nucleus is there. In the generative cell, it is a very small spindle shape sometimes with a very dense cytoplasm and a nucleus, and it floats in the cytoplasm of the vegetative cell. So 60% of angiosperms, the pollen grains are shed at a two cell stage, means vegetative cell and generative cell. But in other generative cells divide the mitotically sometimes. The two male gametes before pollen grains are shed, it is comes down into the three cell stage. So in the 60% cases it is a two cell stage, but in some other cases the two male gametes are again going to the pollen grain to shed into the three cell stage. Next that is the gynoecium. Gynoecium, we know that it is a female reproductive part, or in case of the angiospermic plant it is also known as the pistil, and pistil is the gynoecium innermost word-like structure, and the part of this pistil is the carpel, stigma, style and ovary. If the single carpel, then monocarpellity, many carpels are there, it is the multicarpellity. But each carpel have stigma, the upper portion, style, the long tube-like structure, and the swollen area that is found in the base position, it is known as the ovary. And ovary has many ovules, means megasporangia, that attach to a tissue inside the ovary. If it is going to be attached, so the attachment is known as the placenta, and ovarian cavity is known as the locus. And after that we will see the presentation later on. Number of ovules in a ovary may be one. If the ovule is only one, that is in the wheat, paddy and mango. To many papayas also, watermelons also, very many ovules are there, like in papaya, watermelons, orchids also. Ovary are sometimes single carpel, monocarpellary, and sometimes bicarpellary, and sometimes multicarpellary is there. On the basis of the fusion, if these all the pistils are fused together, many pistils is there, or it is fused together, it is known as the syncarpus, and if it is all free, it is known as the apocarpus. So there is a two position found in the ovarian wall. Now on the basis of this position, what is the position of this relative position of the micropyle actually? The body of ovule and the funicle, if we study, there is a micropyle area and the body, and the funicle is also, that is in the degree of the curvature. What is the degree of this curvature? So on the basis of the degree of curvature, there is the types of ovules, are the 6 type of ovules are found in the angiospermic plants. Like in the case of the if orthotropous, a type of picture is showing the orthotropous, ovule is known as the orthotropous. Here the micropyle, the chalaza and the funicles are in a straight line. Both three are in the straight line. This is the most primitive types of the ovule. It is found in the pepper plant, cycas, polygonum also. So this is very common. And the next one is the anatropous. Anatropous means the ovule turns at the 180 degree angle. It is turns at the 180 degree angle. Thus it is inverted ovule. It is known as the inverted ovule, in which the micropyle lies close to the hilum, and at the side of the hilum it is found in the 90% cases in the angiospermic family. So B type is very common. And second is the campylotropous. In ovules is curved more or less at the right angle to the funicle, and the micropylar end is bent down the slightly. in the member in the leguminous family or the cruciferae in the mustard plants. Here you can find this type. This is the next type. And then it is a C type. So you are going to see that it is the hemianatropous. The ovule turns at 90°. In the anatropous it was 180° angle, but here it is turns at the 90° angle upon the funicle or the body of the ovule, and in a right angle of the funicle. It is found in the Ranunculaceae family, Ranunculus. And next one is the Amphitropous, E type. Ovule is well as entry sac is curved like horseshoe in a lemma or in the poppy plants. In the alisma plants it is found. And the last one is the Sarcinotropous. The ovule turns at the more than 360° angles. You can see here in the F condition, 360° angle. Look at the picture F, 360° angles. It is rotated and the ovule turns at the more than 360° angle. So the funicles becomes coiled around the ovule. It is found in the Opuntia cactus family, and sometimes it is the Plumbaceae family. So in the on the all cactus, mostly the Sarcinotropous type of ovule is found. So this is the anatropous ovule. Mostly 90% cases this type of ovule is found. This ovule, or you can say it is a megasporangium or the female gametophyte. So you can see here, this is the upper portion is the chalaza and lower portion is the micropyle. Lower portion is the micropyle. The funicle or stalk is there. In the center you can see this, there is the egg or the oosphere is there and supported by the two synergids or the helper cells. Secondary nucleus in the center having the two nucleus. It is also known as the polar body, responsible for the development of endosperm, and upper three are known as the antipodals. The integuments is there and nucellus is also there. So this is the anatropous or very common embryo sac found in the angiospermic plants. Now going to see all this part means funicle. What is this funicle? Funicle is that the ovule is attached to the placenta by means of a stalk that is called the funicle, and the hilum, the body of the ovule fused with funicle in the region called the hilum. Hilum, the junction between the ovule and the funicle, it is known as the hilum. There is each ovules have one sometimes and sometimes two protective envelope, that is called the integuments. Inner and outer two integuments is there for the protection of the ovule. Micropyle, the integuments leave small opening at one end, that is called the micropyle. This end is called the micropylar end and the opposite end is known as the chalaza end. So opposite the micropylar end, it is a chalaza that representing the basal part of the ovule. What is the nucellus? Nucellus actually it is a tissue that encloses the embryo sac is known as the nucellus. Nucellus or the tissue. And in the embryo sac is also known as the female gametophyte. You can see an ovule generated has a single embryo sac formed from a megaspore development through the reduction division, through the meiosis cell division, and typically it is nucleated. And what is the nucleate? It is mostly 8 nucleate and 7 cell stage, in which the egg apparatus is there at the micropylar end. It has two synergids and one egg cell and some filiform apparatus that is known as the synergids. It is special thickening at the micropylar end. It guide actually the pollen tube to enter through that end and going to be fused with the egg cell. The antipodals are the 3 in numbers towards the chalaza ends. 2 haploid nucleus at the polar end below the egg aperture. So these are the structure found in the female gametophyte or the embryo sac. So how it is going to be developed? It is known as by the process is known as the megasporogenesis, in the formation of the ovule. Megaspore mother cell, that is known as the MMC, megaspore mother cell. It is actually in the diploid in number, and the cells in the nucleus that is near the micropylar region. After going the meiosis cell division, the megasporogenesis process going to be make the megaspore haploid, mean monosporic development. 4 cells of haploid, 3 nears the micropyles and 1 that is generated, 1 functional, means 3 is going to be degenerate. There in the microsporogenesis, the 4 cells are formed and all the 4s are going to develop into the pollen grain. But here in the 4 cells, the 4 haploid cells, 3 that is near the micropyles, it is degenerated, but 1 megaspore mother cell is the functional. This functional megaspore enlarged and undergoing the mitosis cell division. The 2 nuclei, then 4 nuclei, then 8 nucleus mostly. 2 nuclei moves opposite pole to mitotic divisions, 4 nuclei at the each pole wall formation, and after this the chaligeal end is formed and the micropylar end is formed. Means in the chaligeal end 3 forms the antipodals and micropylar end 3 forms the egg apparatus. Egg apparatus means 1 is the egg and 2 are known as the synergids. The remaining 2 nuclei, that is known as the polar nuclei. It is a very large and combined together to form a central cell. That's why we say that typical megaspore mother cells or typical embryo sac having the 8 nucleus but having the 7 cell structure. This is the megasporogenesis process by the megaspore mother cells, MMC diploids. Here is the micropylar and a nucleus is there. The megaspore mother cells undergoes the meiosis cell division, and after the meiosis cell division, 2 cells, then 4 cells is formed. Micropylar and the megaspore tetrads. The megaspore is going to be tetrads, but 3 having to be degenerated and 1 will be the functional. So you can see here 1 is the functional, and after the functional megaspore mother cells going to the mitotic cell division, so now it is again to form the 4 cells stage, then 8 cells stage, and after formation the 8 cells stage, the functional megaspore enlarges, mitotic division 2 nuclei more opposite towards the poles, to mitotic division 4 nuclei at the each pole, and after that wall formation is there. Only after this stage the chaligeal end 3 forms antipodals, micropylar ends 3 forms egg apparatus, and remaining 2s are going to be inside the 2 polar nuclei. It is also known as the central cell. So this is the stage of the development of the embryo sac in the angiospermic plant. This picture shows a developed embryo sac, means the female gametophyte, that it is known as the polygonum type, very common. You can see here the synergids or the helper cells or cooperative cells is there along with the egg cell. These cells generally possess a micropylar nucleus and a chaligeal vacuole. In the electron microscopic study, we revealed that the synergids lacks a cell wall on their chaligeal side at maturity. They are characterized by the presence of a filiform apparatus of the micropylar tip, and it is in the form of the finger-like projections. Each projections comprises a core of the microfibrils enclosed in a sheet. Usually one synergid start to degenerate just with the pollination. The synergids perhaps secrete some chemotropic substances and thus direct the pollen tube growth inside the embryo sac. So this is the responsibility of the synergids with the filiform apparatus. And in the center you can see a egg or the egg cell. The egg shows the cytoplasmic polarity opposite to the synergids, and its wall is very thick at the micropylar end. Usually the egg has a micropylar vacuole and a chaligeal nucleus. The plasmodesmata connecting it's present in between the eggs and the synergids. And the upper side you can see the antipodals or the vegetative cells. These are vegetative cells of embryo sac. In most of the plants there are 3 antipodal cells, and then this end is known as the chaligeal end mostly. In the central, it is the largest cell of the embryo sac. It initiated contain 2 polar nuclei which fuses just before the fertilization. just before the fertilization means fusion of the male gamete to the egg cells. So just before this event, mostly it fuses just before to form a secondary nucleus, and it is defined as a nucleus that having the 2n number of chromosomes. So this is the typical embryo sac. It is found in the angiospermic plants. This is all about the today's topic. Thank you and